In this video, I'll be summarizing and reviewing this paper concerning intracellular delivery molecules using laser-activated carbon nanoparticles and its potential applications and limitations. The primary focus of the research conducted in this paper is how to effectively deliver drugs, protein, and/or any genetic material into the cell. Because cells typically take in larger molecules through endocytosis, the introduced material undergoes lysosomal degradation, which diminishes the molecule's potency and effectiveness. By molecule, I mean drugs, protein, and the genetic material mentioned above. So, this research proposes direct delivery by cell poration. By exposing injected carbon nanoparticles to nanosecond laser pulses, thermally generated acoustic waves are produced that help break the cell membrane, leading to easy material delivery without significant degradation of the material itself. Multiple mechanisms were explored regarding the photoacoustic emissions, including water vapor bubbles. However, when compared to another experiment with similar conditions, the most sensible mechanism was the carbon steam reaction. Here, the heat produced from the laser pulses react the carbon from the nanoparticles with water vapor to produce hydrogen and carbon monoxide gas. These produced bubbles oscillate and then collapse, releasing an acoustic wave that damages the cell membrane in a manner similar to cavitation. Research was first conducted in vitro using human prostate cancer cells. A mixture of carbon nanoparticles, cell suspensions, and fluorescent markers were irradiated in a rotating laser exposure chamber. Multiple fluorescent markers were used to compare how size of the molecule delivered would affect intracellular uptake. Here, calcine, a common marker used for cell viability, and varying sizes of dextrin, a complex branch glucan, were used for comparison. The laser used in this experiment was a continuum shear light and can be seen in the central figure. The next step was to determine the viability of this experiment in vivo. In this case, the cells from a mouse's tibialis anterior muscle were tested on and analyzed, muscle as indicated in the diagram to the top right. The mice were first anesthetized, then injected with a mixture of carbon black nanoparticles and propydium iodide. The propydium iodide was used to measure uptake. The muscle section was later laser pulsed, and finally the mice were euthanized. The focus parameters in this experiment all concern the effective uptake of the cell, including molecular size and weight of uptake molecule, laser energy, carbon black nanoparticle absorbance, type of nanoparticle, in vitro versus in vivo situations, and lastly, the viability of the cell after uptake. Some techniques used to help analyze the effect of the parameters are flow cytometry, a technique useful for measuring intracellular uptake, laser scanning microscope imaging, used to observe fluorescence, and mass spectroscopy, used to analyze the gas in the laser head chamber. This image is a good representation of laser microscope imaging and was taken from literature. When comparing nanoparticle type results, carbon black nanoparticles were found to induce the greatest efficiency of uptake. Gold nanospheres and carbon multi-walled nanotubes produced insignificant effect and were thus ignored for future result comparisons. On comparison between carbon black nanoparticle size, the laser was set to a 1064 nanometer exposure setting. A linear relationship wasn't observed, however, there was a clear size relation with intracellular uptake. The 25 and 75 nanometer carbon black nanoparticle had similar larger efficiency than the smaller 13 nanometer carbon black nanoparticles, as can be observed in the graphical representation to the right. Carbon black nanoparticle absorbance of photon wavelength from the laser, keeping in mind that the laser essentially is the number of protons per pulse, was also found to have an effect on uptake. The conditions were set to 1 minute exposure time and the laser power from 1 to 50 millijoules per pulse. Decreasing photon wavelength was found to increase the light absorbance of carbon black nanoparticles. Given that absorbance is directly linked with intracellular uptake, this would increase uptake as well, as this figure shows. The wavelengths measured were 1064 nanometers, 532 nanometers, and 355 nanometers. Uptake molecular size and weight also influence the efficiency of uptake. Calcine was found to have the highest efficiency at a mass of 623 daltons and 150 kilodalton dextrin at a little less. On the other hand, at greater than 500 kilodalton dextrins, the efficiency drastically drops. Optimizing efficiency requires under 150 kilodalton molecular weight. Laser energies produced highest uptake at highest energies paired with high carbon black nanoparticle concentrations. Here, concentrations were measured at 15, 30, and 60 milligrams per liter, and energies at 25 to 100 millijoules per pulse. The concentrations in this graphical representation are increasing from left to right, with laser energy as the horizontal axis and uptake as the vertical axis. Finally, although uptake was highest in vitro at around 65% of cells, the high uptake of in vivo cells at 50% is promising and indicates high feasibility of this experiment's success in live bodies from an uptake perspective. In terms of cell viability, in vitro cells were 90% viable and in vivo cells were also highly viable, although the specific percentage wasn't mentioned. In the case of in vivo cells, viability was determined using a nitro-blue tetrazolium assay. The viability and extensive uptake by the cells are promising for future medical applications. The left figure represents the in vitro experiment and the right the in vivo. In figure 1, the left column are treated with carbon black nanoparticles, while the right side is a representation of sham exposures. In figure 8, the left column represents the negative control, and the upper, middle, and lower sections are represented from top to bottom. Not only are nanosecond laser pulses useful for carbon black nanoparticle deliveries, but also are applicable to single-walled carbon nanotube delivery. The single-walled carbon nanotube would itself be the vehicle for delivery instead of simple diffusion, ensuring targeted RNA or DNA vector delivery and controlled drug release for target cells. The acoustic applications of this paper can also be extended to ultrasound or photoacoustic imaging, where laser pulse nanoparticle use can enhance contrast and depth, and when combined with ultrasound, track the location of injected stem cells in vivo. This is extremely useful for tracking cell therapy, identifying diseased tissue, and monitoring treatment of diseased tissue. Although this paper demonstrated successful technical applications to cells, it unfortunately did not address the potential health risks that could be associated with the presence of carbon black nanoparticles, nor did it address carbon black nanoparticle removal methods in case of cytotoxicity from prolonged exposure. Pain observations were also not recorded due to the mice being under anesthesia and immediate euthanasia. Furthermore, there were no noted issues in the literature whatsoever, and the method of cell viability determination was a little unclear. Hopefully, these will be addressed in future or related studies. In conclusion, this paper presents an extremely useful technique for medical applications, but fails to consider any long-term effects and health risks to potential future patients. In the future, it could focus more on long-term studies, potentially be utilized in the aforementioned applications, and focus more on experimental effects on health.